Imagine a world in which what you do at discovery seamlessly scales to clinical manufacturing. That is exactly the future that Kytopen is enabling. And today you will hear about our exciting Flowfact non-viral delivery technology that has that capability, using CD34 stem cells as the use case for mRNA delivery optimization and scale up. I am Paulo Garcia, CEO and co-founder of Kytopen, an MIT spinout that resides at the Engine in Cambridge, Massachusetts. And what we're working on is in innovating in the delivery of genetic material into cells in a completely non-viral manner. There are more than 7,000 rare diseases in the world, and many of them are life-threatening and are affecting hundreds of millions of patients in the world. This means that there is an opportunity to help these patients from the suffering that they are imparting by innovating in treatments and future generation type of cell therapies. What is exciting for all of us is the fact that we are in the middle of a biotech revolution that is enabled by many of you, therapeutic developers and academic pioneers, that are advancing cell therapies in order to cure and treat many of these devastating diseases. At a high level, cell therapy manufacturing uses patients' own cells. They get genetically modified, they get expanded, and then they get reinfused into a patient. This is an actual really complex process that is generating some tremendous promise in the field. And that the field is bright. There are several approved therapies in which this process is being perfected, is being advanced, and it is helping patients that are suffering from these diseases. Some of the limitations, though, are in the fact that they are still cost prohibitive. They are hundreds of thousands of dollars or millions of dollars per dose, and the manufacturing is variable. So not every patient is benefiting from these life-saving therapies. At Kytopen, we are developing technology to enable the tomorrow's approved therapies that will be even more complex. They will require multiple gene edits. They will require delivery of large payloads into cells. And more importantly, for them to be able to reach the patients, they need to become more cost effective and with more consistent manufacturing. The step of introducing the genetic material into the cells is a critical step and one of the major bottlenecks in the industry. Kytopen is tackling that problem, the ability to deliver genetic material in the form of mRNA, DNA, and CRISPR-Cas RNP to cells that are being developed for these therapies, such as T cells, CD34 stem cells, and NK cells. The reason we're doing that is we want to unlock the bottlenecks that we have observed in the industry, which are generally that the discovery is being held hostage by the clinical manufacturing, the delivery of these life-saving therapies into patients. And with technology, we believe we can break this barrier. In addition, by providing technology that is the same at discovery and at delivery scale, we believe we can help accelerate the field. And finally, imagine if you could do clinical grade discovery, meaning whatever technology you're using at discovery seamlessly scales to your clinical manufacturing to open up the doors for new opportunities. That is Kytopen. And Kytopen is focusing on combining the discovery and the delivery process into one single solution that can be used at preclinical, clinical, and commercialization scale, specifically to enable you, therapeutic developers, to accelerate into the clinic and help many more patients. Let me tell you how we're actually doing this. We have a proprietary technology that we call Flowfact that is patent protected by 5 families of pending patents, in which we are combining two types of energies in order to disrupt the outer and the inner membranes of the cells and deliver genetic material all the way into the nucleus. The mechanical energy is imparted by the continuous fluid flow that we are using in our channels. In addition, we expose the cells to low energy electrical pulses in order to facilitate in the delivery of the genetic material. If we only use our mechanical energy, we do not get delivery. If we only use our low energy electrical fields, we do not get delivery either. It's only when we combine these two types of energies that we can actually efficiently deliver genetic material into cells while retaining their viability and their functionality. And this has opened up the doors for the technology to really be impactful, not only at discovery, but also for clinical manufacturing, as you will see. We have designed and built our first generation beta device of the technology. This is a closed system that can process hundreds of milliliters of input material in a continuous manner through our cartridge that exposes the cells to the right flow effect conditions and directly into the output vessel. In this case, it is a bag that contains the growth media. The channels that we are employing process many cells at any given amount of time. This facilitates the throughput, and in that channel is where the cells experience the mechanical energy from the fluid flow and the electrical energy from the pulses that we program. We have outfitted a liquid handling system so that we can do high throughput process development optimization at low volumes. Here we're aspirating the sample that contains the cell and the payload of interest. When we dispense the cells into growth media, that's when we expose the cells to the right parameters of flow effect. And we can use that as a screening tool. What you see here is a 96 well plate that can be processed in less than 10 minutes with unique combinations of flow rates and electric fields. We can identify the superior performance condition and then pour that directly into our large volume manufacturing so that our partners actually can scale up in a seamless manner. That single flow cell in the center is identical at the discovery scale and at the clinical manufacturing scale without any modifications. We use the same flow rates, the same electric fields, the same materials, the same buffer. Nothing changes. The only thing that is adjusted is the amount of time that it takes to process these samples. So we have eliminated virtually the scale up time because of the continuous flow nature of our process. As a case study, we will be talking about the CD34 stem cell optimization that we took from optimization all the way to scale up in a time frame of 6 weeks, in which we implemented culturing protocols. We did a design of experiments for screening the flow effect conditions, which was then confirmed at different number of cells and scales that were then confirmed in the 50 million range of cells. What's important is that we're not compromising the functionality of these cells, and we are achieving viabilities, efficiencies, and yields that are all greater than 90% in this very fragile population that is being developed for therapeutic purposes. CD34 stem cells. That is the case study that I'm going to be sharing with you today. On the left, we see flow cytometry plots in which we have introduced the cells into our proprietary buffer, our transfection buffer, and the cells exhibit a particular morphology. What's exciting for us is that the changes that are seen with other non-viral technologies are not evidenced here. Those transfected cells with the Flowfact technology actually remain intact. And this is at 24 hours after the cells have been transfected with an mRNA payload that expresses green fluorescent protein or GFP. Let me walk you through the actual experimentation in which we used CD34 stem cells. We transfect them with mRNA that encodes GFP. And for these initial studies, we used 250,000 cells per well. The analysis was conducted at 24 hours. What you're seeing here is a heat map. This heat map represents what the 96 well plate geometry is able to accommodate. On the y-axis, we have a metric of waveform, which is a surrogate for the amount of electrical energy that we introduced, A being the highest energy and I being the lowest energy. On the x-axis, we have the flow rate, 1 being the lowest flow rate, 7 being the highest flow rate that we studied. And what this means is that we have a really broad dynamic range of flow effect parameters that we can actually study. What we're showing here is the cell viability of cells that were processed with our technology. And you see that G, H, and I all exhibit very, very competitive cell viabilities of greater than 90%. So viability, we know exactly where to look for the right parameters. Efficiency, which is another critical metric, is also studied here, in which we have a majority of the plate with greater than 90% transfection efficiency as measured via flow cytometry by the expression of the GFP. When we combine the viability and the efficiency, we can actually identify where the yield is maximized. And that is at that G7 area in which both are maximized and we are achieving greater than 90% yields. These high efficiencies are observed across a majority of conditions, and this really enables us as an initial screen to then go and confirm the initial outputs that we achieve with our high throughput capabilities. So what we did is we ran these samples now with replicates, specifically in triplicates, in order to confirm the results that we had seen earlier. What we're seeing here is, again, the viability, the efficiency, and the yield of the 3 cell numbers that we processed in our technology. The orange bars are the 250,000 cells per well. We actually decided to go down in cell number in order to accommodate a cell population that could be very limited, particularly with patients that are being treated with different therapeutic regimes. That is the pink bar, the array in which we are processing 100,000 cells per well, and the dark blue in which we're processing 20,000 cells per well. These are very low cell numbers, and what we're demonstrating here is not only the ability of our technology to process low cell numbers, but also the fact that the results are consistent. The viabilities are greater than 95%, the efficiencies are greater than 90%, and more importantly, the yield, the number of live cells that are successfully engineered are greater than 100%. And this means it confirms that our process is extremely gentle to the cells, and that in a case in which you needed to maximize the use of precious sample, you can actually go down to those cell numbers. But it didn't stop there. We did a scale down. We also demonstrated a scale up, leveraging the capabilities of our technology that only require more time to process more cells. What you see here is, again, the viability, the efficiency, and the yield of the low volume array platform with 250,000 cells in the orange bars and 50 million cells in the light blue. They resemble the viability, the efficiency, and the yields, again, exceeding 100% at 24 hours, because these cells retain their capability of cell division. They are expressing GFP, and this is really exciting for the industry in that we can process those 50 million cells in a volume of 20 milliliters in less than 30 seconds. I'll repeat that. We processed 50 million CD34 stem cells in a volume of 20 milliliters in less than 30 seconds. And this really provides opportunities for a broader range of discovery and clinical applications that can be eventually implemented for helping many more patients that are suffering from many diseases. The Flowfact technology links target discovery, process development optimization, and clinical manufacturing in a single gene delivery technology, the flow cell that you see on the left. It can be used for research scale discovery. It can be used for IND studies in the preclinical setting. It can be used in the clinical setting to identify the safety and efficiency profiles for patients. And eventually, it can be used for commercial delivery of these life-saving therapies to many patients. Our technology scales seamlessly across all of these different stages of development, again, for discovery, screening, and delivery of the life-saving therapies for patients that are suffering from many diseases. Why? Why? Because Kytopen employs continuous fluid flow from discovery scale to clinical manufacturing, and all that we have to do in order to scale is to flow for a longer period of time. For example, a 100 milliliter volume can be processed in 2 minutes, and those 2 minutes will enable you to process 5 billion cells. But what if you wanted to process 500 milliliters with 25 billion cells? You can also do that. You can do that by just flowing for 10 minutes. That is the type of scale that Kytopen enables with our Flowfact technology, and leveraging the fact that we can do target discovery and process development optimization in a high throughput manner using low volumes in less than 10 minutes. We're Kytopen, and we are bringing together the next generation non-viral cell and gene therapy discovery and delivery technology to redefine what is truly possible and enable you, therapeutic developers and academic pioneers, from getting a head start on the future of the industry, being able to identify the next generation cell therapies that will be curative or that have the potential to be curative and help many, many patients suffering from these rare diseases. So thank you for your time. Looking forward to connecting with many of you during the partnering sessions and wishing you a great meeting on the Mesa conference. Thank you.